Consider this. We can send rovers to Mars. We can edit our own genes. We've even mapped the entire human genome. Yet, after more than 40 years of intense worldwide scientific effort, a complete cure for HIV remains just out of our reach. It's a puzzle that has stumped some of the brightest minds on the planet. Why is this one virus so incredibly difficult to defeat? You might think that with all our modern medicine we would have figured it out by now. It's a question that deserves a really good answer, and the answer is truly fascinating. It's not just one thing, it's a combination of clever tricks the virus uses to survive. The story of HIV is a story of a microscopic battle happening inside the human body. It's a high-stakes game of hide-and-seek played at the cellular level. Imagine trying to catch a spy who can not only become invisible, but can also change their face and change their uniform in an instant. That's what our doctors and scientists are up against. This virus doesn't play by the rules, so let's get into it. So, what exactly happens when HIV enters the body? Let's dive in. Our bodies have an amazing defense force called the immune system. A key part of this force is a type of white blood cell called the CD4 T cell. Think of these CD4 cells as the quarterbacks of your immune team. They call the plays. They identify threats. They direct other immune cells to attack and destroy invaders like bacteria, other viruses. They are absolutely essential for keeping you healthy. When you get a cold, it's your CD4 cells that rally the troops to fight it off. They are the heroes of your internal world. HIV is incredibly sneaky because it specifically targets these CD4 cells. It doesn't just fight them, it takes them over. The virus has keys on its surface that match locks on CD4 cells. One of these locks is the CD4 receptor itself. But to get inside, HIV usually needs a second key, a co-receptor, CCR5, CXCR4. When the virus uses its keys to unlock both doors, it breaks into the cell. It's a specific two-step security breach that gives HIV access to the cell's command center. Once inside the CD4 cell, HIV unleashes its master plan. As a retrovirus, it carries an enzyme called reverse transcriptase. Now, you might be thinking, if the virus is in the blood, can't our medicines just find it and destroy it? That's a great question. It leads to one of the biggest problems in finding a cure. HIV is a master of hiding. While many virus particles are active in the bloodstream, some go into a dormant sleeping state. They infect a CD4 cell and integrate into the cell's DNA, then they just stop. They don't make new viruses. They sit silently waiting. This creates what scientists call a latent reservoir. These reservoirs are like secret sleeper cells scattered throughout the body, lymph nodes, the gut, the brain, other tissues where they're shielded from the immune system and our strongest drugs. Antiretroviral therapy, or RT, is fantastic at stopping the active virus. It prevents HIV from making new copies, letting the immune system recover. But the drugs can't tell a healthy cell from one silently harboring virus. Drugs only work on viruses that are actively replicating. So even when someone on treatment has an undetectable viral load in their blood, the latent reservoir remains. The virus isn't gone, it's in hibernation. If they stop taking medication, sleeping cells can wake and start producing virus again. As if hiding wasn't enough, HIV has another incredible trick up its sleeve. It mutates or changes at a breathtakingly rapid pace. Think of it like a spy who not only has a perfect disguise, but can also change their face every few minutes. The enzyme we talked about earlier, reverse transcriptase, is very sloppy. When it copies the virus's genetic code, it makes a lot of mistakes, about one error for every new virus particle it creates. In most living things, these kinds of mistakes would be bad. But for HIV, this sloppiness is its greatest strength. Every day, a person with active HIV produces billions of new virus particles. And because of the sloppy copying, many of these new viruses are slightly different from the original. They have tiny changes in their genetic code and, as a result, in their outer structure. This means the immune system is constantly playing catch-up. Just when it learns to recognize one version of the virus, a new, slightly different version appears that it doesn't recognize. It's like trying to hit a target that is constantly moving and changing its shape and changing its color. This is called viral diversity. Now for something truly amazing. In this global battle against HIV, scientists discovered something extraordinary. Some people seem to be naturally immune to the virus. They can be exposed to HIV, but they just don't get infected. For years, this was a medical mystery. How could this be possible? The answer, it turns out, lies in their genes. Remember those co-receptors, the second locks that HIV needs to pick to get inside a CD4 cell? The most common one is called CCR5. It acts as a doorknob that the virus grabs onto to pull itself into the cell. Scientists found that these naturally resistant people have a specific genetic mutation. It's called the CCR5 delta 32 mutation. This isn't a disease or a disorder, it's simply a genetic variation. People with this mutation have a slightly shorter, non-functional version of the CCR5 protein. Because of this change, the CCR5 co-receptor isn't present on the surface of their CD4 cells. So when HIV comes along and tries to infect their cells, it finds the main door, the CD4 receptor, but the doorknob, the CCR5 co-receptor, is missing. The virus can't get a grip to pull itself inside. The door is effectively sealed shut. This genetic trait is most common in people of Northern European descent. It's estimated that about 1% of this population has 2 copies of the CCR5 delta 32 mutation, which gives them this powerful resistance to the most common strains of HIV. The discovery of the CCR5 delta 32 mutation wasn't just a cool scientific fact, it sparked a revolution in HIV research. Scientists thought, if nature can make a person resistant to HIV by removing the CCR5 receptor, can we do the same thing for someone who already has the virus? This idea led to some of the most groundbreaking experiments in the history of medicine. It shifted the focus from just controlling the virus to potentially eradicating it from the body entirely. This natural immunity provided a roadmap for a functional cure. This roadmap led to the incredible cases of the Berlin patient, Timothy Ray Brown, and later, the London patient, Adam Castillejo. Both men had HIV and also developed a type of blood cancer that required a stem cell transplant. Doctors had a brilliant idea. What if they found a stem cell donor who had 2 copies of the CCR5 delta 32 mutation? The transplant would not only treat the cancer, but also replace the patient's entire immune system with a new one that was naturally resistant to HIV. It was a risky and complex procedure, but for both men, it worked. They were cured of HIV. These cases proved that a cure for HIV is possible, and with science leading the way, a future without HIV is not just a dream, it's a destination we are getting closer to every single day.